A very good day to each and every one. It is my absolute joy and great pleasure to welcome you all once again to Alnasacademy for part 11 of chapter 1, Reproduction in Lower and Higher Plants, Class 12, Biology, Revised Syllabus 2020 of Maharashtra State Board, which also covers NCERT Unit 6 Chapter 2. And I am your facilitator, Nusrat Siddiqui, to help you and to guide you. Let's begin our video with yet another inspiring quote by Sidney J. Harris. The whole purpose of education is to turn mirrors into windows. This means that education opens a window near and far throughout and let us understand the world in a deeper and more complex way. Let us understand our today's topic, that is double fertilization. We have already learnt about pollen-pistil interaction in our previous video, which leads to our next topic, that is double fertilization. In our previous video, we have studied about artificial hybridization. Then we have discussed about pollen-pistil interaction, and pollen-pistil interaction, it starts with pollination and it ends with fertilization. Now in pollination, the first step is identification of pollen grains, which leads to the secretion from the stigma, mainly sugar, as nutrients for the pollen grains for the pollen tube to germinate. So once the pollen grains are identified and right or compatible pollens are identified, the stigma, it secretes the nutrients, mainly sugar, for the pollen grain to absorb, and after the secretion of the sugar from the stigma, the germination of pollen tube takes place. After the pollen grain has reached the surface of the stigma, it germinates and it forms the pollen tube, which penetrates the stigma, style and ovary chamber and then enters the ovule. Now the growth of pollen tube is guided by the filiform apparatus and it enters one of the synergid. So over here, this is the filiform apparatus of the synergid, which guides the pollen tube towards itself. There are three possibilities of pollen tube entering the ovule. If the pollen tube enters through the chalaza, then it is termed as Chalazogamy. So over here you can see the pollen tube is entering through the chalaza, and the pollen tube which you are seeing over here are with the callose plugs. This is the diagrammatic representation for you all for better understanding how the pollen tube enters through the chalaza. The second possibility is that in some of the plants, the pollen tube enters by piercing the integument and it is called as Mesogamy. So pollen tube over here you can see it is piercing through the integument and then it is entering the embryo sac through the micropylar end. And the third possibility, and usually the pollen tube enters through the micropyle and it is called as Porogamy. So if pollen tube enters through the chalaza, it is called as Chalazogamy. If it pierces through the integument, then it is called as Mesogamy. And if it enters through the micropyle, it is called as Porogamy. And here we have taken an example of most common ovule, that is anatropous ovule. The pollen tube carrying male gametes penetrates in one of the synergids. Over here, the watery contents of the synergids are absorbed by the pollen tube, which then ruptures and releases the content. Now this content is nothing else but these are two non-motile, that is non-movable male gametes. As the male gametes are non-motile, they cannot move, the pollen tube carries them. This process is called as Siphonogamy. So Siphonogamy is the process where male gametes are carried by the hollow pollen tube, and it is to ensure fertilization. So it is a condition where plant develop a hollow pollen tube which carries male gamete, and this male gamete then later on fuses with the egg to form the zygote, and this is to ensure fertilization. So Siphonogamy is nothing else but it is the process where male gametes are carried through the hollow pollen tube. Let us do a quick recap of the embryo sac and the cells which are present in it. So at the chalazal end there are three antipodal cells. Then in the center there are two polar nuclei, and towards the micropyle end there are two synergids with the filiform apparatus and one large cell which is the egg. Also all these cells are haploid in nature. Now we are focusing only on the embryo sac, and this is the diagrammatic representation of the embryo sac. Of the two synergid, one of the synergid will degenerate and the filiform apparatus of the degenerating synergid will guide the pollen tube to enter into the embryo sac, and it will degenerate. The wall of the synergids will degenerate, and now the two male gametes are in the embryo sac. The pollen tube and the tube cell, it ruptures at this point, releasing the two male gamete in the cytoplasm of the synergid. Over here, the fertilization will now start. So one of the male gamete will fuse with the egg, forming a diploid zygote, and this process is known as syngamy. So male gamete is haploid, the egg is also haploid, thus the fusion of two haploid cells results in the formation of zygote. So one of the male gamete will fuse with the egg, forming a diploid zygote, and this process is known as syngamy. In the second fusion, the second male gamete will fuse with the polar nuclei, forming a triple fusion and forming a triploid endosperm. So polar nuclei, they are also haploid. So there are two polar nuclei. So two haploid polar nuclei fusing with one haploid male gamete. So three haploid cell fuses together. That's the reason why it is termed as triple fusion. And this type of fusion, the primary endosperm nucleus is formed, and it is also referred to as PEN. P for primary, E for endosperm, and N for nucleus, and the process is known as triple fusion. So the second male gamete fuses with the polar nuclei to form triploid endosperm, which is also termed as PEN. Later on, the primary endosperm nucleus forms primary endosperm cells. So over here we see that syngamy is a generative type of fertilization, whereas triple fusion is a vegetative type of fertilization, where it provides in triple fusion, the primary endosperm nucleus, it provides nourishment. It forms the nutritive tissue and provides nourishment to the developing embryo, which is formed from the zygote. So zygote will later on develop into an embryo, and it will gain its nourishment from the endosperm. So over here we see that both the male gametes participate in the fertilization. That's the reason why it is called as double fertilization. Let us understand the sequence in double fertilization. First, formation of zygote will take place, which is followed by formation of primary endosperm nucleus, then development of endospermic tissue, which is a nutritive tissue, and then the development of embryo. So over here, first the zygote formation is important. So once the zygote is formed, primary endosperm nucleus is formed. Now this primary endospermic nucleus, it provides nourishment to the developing embryo. So after the primary endosperm nucleus is formed, it develop into the endospermic tissue, which provides nourishment to the embryo. So after the formation of nutritive tissue, embryo development starts. So the sequence will be formation of zygote followed by primary endosperm nucleus. This primary endosperm nucleus will form endospermic tissue, which is a nutritive tissue, and then development of embryo will start. Fertilization was first discovered by Strasburger in 1884. Double fertilization, which is a complex fertilization mechanism in flowering, that is angiospermic plant, was first discovered by Navastrin in liliaceous plant like Lilium and Fritillaria. Let us understand the significance of double fertilization. So it is the unique feature of angiosperm. Then it ensures that parent plant invests a seed with a food store. So in a seed there is a food store, and this will happen only if the egg is fertilized, and the diploid zygote then develops into the embryo, which consequently develops into a new plant. It restores the diploid condition by the fusion of haploid male gamete with haploid female gamete through the process of syngamy. So we know that the male gamete was also haploid and the female gamete, that is egg, was also haploid. So both these haploid cells, they fuse together to form a diploid. Okay, and this is through the process of syngamy. Now through the double fertilization, it also avoids polyembryony. Polyembryony means formation of more than one embryo from a single fertilized ovum. This we are going to study further in detail in our upcoming video. Next, the triploid endosperm which is formed, it develops into nutritive endosperm tissue and it provides nourishment to the developing embryo. Let us quickly do a recap as to what we have studied in this particular video. We have discussed about the three possibilities of the pollen tube entering into the ovule. The first one is Chalazogamy, when the pollen tube enters through the chalaza. The second one is Mesogamy, when the pollen tube penetrates into the integument. And the third one and the most common one is Porogamy, where the pollen tube enters through the micropyle. Then we have discussed about Siphonogamy. So Siphonogamy is a process where the pollen tube is formed and it's an hollow pollen tube which is formed to carry the male gamete to the ovary in order to ensure fertilization. Next we have studied about Syngamy. Syngamy results in the formation of zygote, and Syngamy is the fusion of haploid male gamete with the haploid egg in order to form diploid zygote. We have also studied about triple fusions. In triple fusion, the polar nuclei fuses with the male gamete to form primary endosperm nucleus, which is the nutritive tissue for the embryo. We have discussed about double fertilization and why the process is termed as double fertilization, because both the male gamete participate in the fertilization, one forming a zygote and second fertilization forms primary endosperm nucleus. That's the reason why it is termed as double fertilization. And lastly, we have discussed about the significance of double fertilization, that is it prevents polyembryony, and there are many other significance of double fertilization which we have discussed in this particular video. That's all for today. Thanks for watching this video. If you have any questions, do write to me at alnasacademyofficial@gmail.com. So till we meet next time, stay safe, stay positive, do like, subscribe and share this video with your friends so that even they may benefit. An entire playlist will be uploaded soon. So till we meet next time, take care, bye bye, thank you and happy learning.